Today we are going to learn about staining. The first step in the staining procedure is preparation of a smear. Here most initial observations of the microorganisms are made with stain preparations. So staining simply means coloring the microorganisms with a dye that emphasizes certain structures. So before the microorganisms can be stained, they must be fixed first. Here fixing simultaneously kills the microorganisms and attaches them to the slide. So it also preserves various parts of the microbes in their natural state with only minimal destruction. When a specimen is to be fixed, a thin film of a material containing microorganisms is spread over the surface of the slide. This film is called smear, which is allowed to air dry. In most times, stain procedure is done by passing the slide through a flame of Bunsen burner several times. Smear slide up or by covering slide with a methyl alcohol for 1 minute. Air dry and flaming fixes the microorganism to the slide. Okay. Then the stain is applied and then washed off with water. Stain is blotted with absorbent paper. Without fixing the stain might wash the microbes off the slide. So the stained microorganisms are now ready for the microscopic examination. This is how we will prepare the smear and stain the smear. Here stains are salts composed of a positive and negative ions, one of which is colored and is known as chromophore. The color of so-called basic dyes is in the positive ions. In acidic dyes, it is in negative ion. Bacteria are slightly negatively charged at pH 7. Thus, the colored positive ions in the basic dye is attracted to the negatively charged bacteria cells. Example for basic dyes includes crystal violet, methylene blue, malachite green, safranin. So, the acidic dyes are not attracted to the most type of bacteria because the dyes negative ions are repelled by the negatively charged bacteria surfaces. So the stain colors the background instead. Preparing a colorless bacteria against a colored background is called negative staining. So it is used in observation of cell shapes such as capsule because the cells are visible against a contrast background. Distortion of cell size and shapes are minimum because heat fixing is not necessary. Cells do not pick up stain. Example of acid dyes are eosin, acid fustian, nigrosin. In order to apply acidic or basic dyes, microbiologists use three kinds of staining techniques. What they are means simple staining, differential staining, special staining. So here differential staining is again of two types, that is gram staining and acid fast staining. Special staining is used for to study the capsules, spores and flagella of bacteria. First one is simple staining. Here simple stain is an aqueous or alcoholic solution of single basic dye. The primary purpose of a simple stain is to highlight the entire microorganism so that the cellular shapes and the basic structures are visible. Stain is applied to the heat fixing smear for a certain length of time and then washed off and the slide is dried and examined. Occasionally a chemical is added to the solution to identify the stain, such as additive is called mordant. So function of a mordant is to increase the affinity of a stain for the biological specimen. Another is to coat a structure to make it thicker and easier to see after it is stained with a dye. Simple stains used in laboratory are methylene blue, carbon fustian, crystal violet, safranin. And next staining is differential staining. So unlike simple stains, differential stains react differently with different kinds of bacteria and thus can be distinguished among them. Differential stains most frequently used are gram staining and acid fast stain. First one is gram stain. Gram stain was developed by the biologist Hans Christian Gram. It is one of the most useful staining procedure because it classifies bacteria into two large groups, gram positive and gram negative. Procedure. A heat fixed smear is covered with a basic purple dye, usually crystal violet. Because the purple stain imparts its color to all cells, it is referred as a primary stain. After a short time, the purple dye is washed off and the smear is covered with iodine, a mordant. When iodine is washed off, both gram positive and gram negative appears dark violet or purple. Next, the slide is washed with alcohol or an alcohol acetone solution. This solution is a decolorizing agent which removes purple from cells of some species but not from other. The alcohol is rinsed off and the slide is then stained with safranin, a basic dye. The smear is washed again, blotted dry and examined under microscope. So here what happened means the purple dye and the iodine combines in the cytoplasm of each bacteria and color it dark violet or purple. The bacteria that retains this color after the alcohol has attempted to decolorize them are classified as gram positive. Bacteria that lost the dark violet or purple color after decolorization are classified as gram negative. So because gram negative bacteria are colorless after the alcoholic wash, they are no longer visible. This is why the basic dye safranin is applied. So it turns the gram negative bacteria to pink color. Here stains such as safranin that have a contrasting color to primary stain are called counter stains because gram positive bacteria retains the original purple stain. They are not affected by the safranin counter stain. So different kinds of bacteria reacts differently to the gram stain because structural differences in their cell walls affects the retention or escapes of a combination of a crystal violet and a iodine called crystal violet iodine complex. So among other differences, gram positive bacteria have a thicker peptidoglycan cell wall than a gram negative bacteria. In addition, gram negative bacteria contains a layer of lipopolysaccharide as a part of the cell wall. So when applied to both the gram positive and gram negative crystal violet and the iodine, readily enters the cell wall. So here inside the cells, the crystal violet and the iodine combines to form a crystal violet iodine complex. This complex is larger than the crystal violet molecules that enters the cell, and because of this size, it cannot wash off intact peptidoglycan layers of a gram positive cells by alcohol. Consequently, gram positive cells retains the color of crystal violet dye. So in gram negative cells, however, the alcohol wash disturbs the outer lipopolysaccharide layer and the crystal violet iodine complex is washed off through the thin layers of the peptidoglycans. As a result, the gram negative cells are colorless until counter stained with safranin, after which they are pink. Summary: here gram positive cells retain the dye and retains purple. Gram negative cells do not retain the dye. They are colorless until counter stained with red colored dye, after which they appear pink. Clear? Next, gram reaction of a bacteria can be provided valuable information for the treatment of diseases. Gram positive bacteria tends to kill easily by the penicillins and cephalosporins. Gram negative bacteria are more resistant because the antibiotics cannot penetrate the lipopolysaccharide layer. Next stain is acid-fast stain. Another important differential stain is acid-fast stain, which binds strongly only to the bacteria that have a waxy material in their cell wall. Here microbiologists use this stain to identify all the bacteria in the genus. Mycobacterium includes two important pathogens, that is Mycobacterium tuberculosis, Mycobacterium leprae, also used to stain genus Nocardia. Procedure: in acid-fast staining, a red colored dye, that is carbylfuscin, is applied to the fixed smear and the slide is gently heated for several minutes. That is, heating enhances the penetration and the retention of a dye. For this purpose only we are used to heat. Then slide is cold and washed with water. The smear is next treated with a acid alcohol, a decolorizer, which removes the red color stain from the bacteria that are not acid fast. The acid fast microorganisms retain red color because the carbylfuscin is more soluble in cell wall lipids than in the acid alcohol. In non-acid fast bacteria whose cell walls lack the lipid components, so the carbylfuscin is readily removed during the decolorization, leaving the cells colorless. The smear is then stained with methylene blue counter stain. The non-acid fast cells appear blue after application of a counter stain. Special stains. These are used to color and isolate the specific parts of microorganisms such as endospores, flagella, capsules. Negative staining of capsule. So many microorganisms contains a gelatinous covering called capsule. In medical microbiology, demonstrating the presence of a capsule is a means of determining the organism's virulence, the degree to which a pathogen causes a disease. Okay, the capsule staining is more difficult than other types of staining procedures because the capsular materials are soluble in water and may be removed during rigorous washing. To determine the presence of a capsule, a microbiologist can fix the bacteria in a solution containing a fine colloidal suspension of colored particles, usually Indian ink or nigrosin, to provide a dark background, and then stain the bacteria with simple stains such as safranin. Because of their chemical composition, capsules do not accept most biological dyes such as safranin and thus appear as halos surrounding each stained bacteria cells. So here from the diagram we will see how the capsules get stained. Next, endospore staining. An endospore is a special resistant dormant structure formed within a cell that protects a bacterium from adverse environmental conditions. Endospores cannot be stained by ordinary methods such as simple staining, gram staining because dyes do not penetrate the walls of the endospore. So most commonly used endospore stains is Saffir-Fulton endospore stain. Malchite green, the primary stain is applied to the heat fixed smear. Heated to steaming for about 5 minutes, heat helps the stain penetrate the endospore wall. Then the penetration is washed for about 30 seconds with water to remove all malchite green from all cell parts except endospore. Next, safranin, a counter stain, is applied to the smear to stain the portion of cells other than the endospore. In a properly prepared smear, the endospores are highly refractive. They can be detected under light microscope when unstained, but they cannot be differentiated from inclusions of stored materials without a special stain. Next staining is flagella staining. Bacteria flagella, in singular form it is flagellum, or the structures of locomotion, too small to be seen with a light microscope without staining. A tedious and a delicate staining procedure used a mordant and a stain carbon fustian to build up diameters of a flagella until they become visible under light microscope. So microbiologists use the number and the arrangement of flagella as a diagnostic aids. So this is about various staining processes that are used for the identification of various microorganisms. Thank you for watching this video. Please subscribe to our channel for more videos.